Hi, it's Mrs. Scoville, and I'm back today to talk to you about the cell cycle. We're also going to be talking about how this relates overall to cell division, which is just our overall new unit. We have 3 learning objectives for this lesson. The first is to explain why cells actually go through the cell cycle. The second is to be able to identify all the stages in the cell cycle, and finally to explain how this is regulated, the cell cycle. So the first part, our first objective here, is this idea of cell growth. So I think you know when you're a baby, you're much smaller than you are now, and this is because all living things grow basically by making more cells. Now you might be thinking, well, couldn't they just get bigger if the cells get bigger? Yes, up to a point, cells can get bigger, but if they get too big, they have to divide or become 2 cells. And this happens because as cells get bigger, their surface area is also increasing, but it's going to increase at a much faster rate than it's going to increase at a much smaller rate than the volume inside is increasing. So what this is going to cause is some real problems in the cell in terms of moving waste and nutrients around. So this can mean that you have too much waste and it takes too long to get to the cell membrane to get out, and that's obviously a problem. It also can mean it takes too long to get nutrients into the cell where it's needed. So this in itself is a problem, and it causes the cell to divide. But furthermore, there also isn't enough DNA really to run the cell when it gets to be too big. So you can see here that cell is very large relative to the other ones. Even when the cell gets bigger, there's not more DNA, so this does become a limiting factor as well. So again, things grow by making more cells. Have to divide once they get too big. So during the cell's growth cycle, a couple of things are going to happen. First, it is going to grow, and then it will divide. All the events that happen leading up to that division and that period of growth we call the cell cycle. So here, the technical definition: series of events that cells go through as they grow and divide. Like I said, there's going to be several things that happen. So the first is the cell's going to grow. And we know that when it gets too big, then it's going to have to divide, so it will prepare for that division. And then finally, it will divide to form 2 what are called daughter cells. So just like mom and a dad have a daughter, it's the same type of thing here. You have one cell that makes 2 daughters, and each of those can then begin the cell cycle themselves. So you can see this goes on and on and on and on, and this is how things stay alive. So our cell cycle itself is going to be divided into 4 phases. Okay, so the first is called G1, or a gap phase. It's the first one because it's G1. And the next one is called the S phase, or the synthesis phase. This is going to be where DNA is synthesized. G2 is next. It's the second gap phase. And finally, you have the M phase, which is mitosis and cytokinesis. And we're going to go through each of these individually. One thing I do want to point out to you here, these first 3, G1, S, and G2, make up a broader category called interphase. So we can look at this in terms of kind of almost like a modified pie graph, but it is a cycle, so it is a circle, so this is an appropriate way to see it. So you can see G1, S, and G2, those all make up interphase, and we're going to go through each of these individually again, but G1, that's mainly where the cell is growing and getting bigger. In S phase, that's synthesis, DNA is being replicated, and that makes sense. You've got to copy it so that each cell gets some. And then finally G2, that second growth phase, this is mainly where it's preparing to go into mitosis and cytokinesis, which we broadly call cell division. So this kind of diagram, you need to be quite familiar with looking at it. Okay, so now we're going to go into first interphase, because as you saw from our graph, if we think of this as time, the cell is going to spend the majority of its time in interphase. And this is the period of growth that occurs between the cell divisions. So again, mitosis and cytokinesis together. And again, there are those 3 phases, so G1, S, and G2. And the G stands for gap, and then the S stands for synthesis. So we're going to look at each phase individually now. So first G1, and just a reminder, G1 equals gap 1, like I just said. And the main thing that happens here is the cell is going to increase in size. So a couple reminders here, like we already saw, when the size of the cell gets too large, this is going to prompt the cell to really have to divide, because that surface to volume ratio really becomes too small. So that means there's too much volume inside, not enough surface outside. The consequence of that is that materials like oxygen cannot diffuse well into the cell, also sugar, and also bad things can't get out, like CO2 can't get out. This can kill the cell, so this is very important that that ratio is appropriate. This is also going to be a place where new proteins and organelles that are going to be needed later are synthesized. So a lot of the organelles get copied at this point. This is also going to be the longest phase of the cell cycle, as you probably saw on the diagram. We can look back at that. S looks a little bit long here too. That depends on the actual cell, but G1 is usually the longest. Okay, so now the next one, the S phase, remember that stands for synthesis. A reminder, synthesis means something is being made. Well, the thing that's being made here is DNA. So DNA gets replicated, and this picture here is showing you some chromosomes. And what's going to happen is these are what are called chromatids. These are individual chromosomes, and those get copied, okay, during the S phase. So this ends up giving you a double copy of that individual chromosome, so that later each cell will get one. All right, the last phase and interphase is G2 or gap 2. And here there's a few more organelles that are going to get made, and also a lot more molecules and proteins. But these are going to be things that are pretty much just needed for the cell to divide. So once we get more into mitosis, we'll focus on what those organelles are. But remember, most of the cell organelles were made back in G1. Finally, it is important to note that once G2 has been completed and all these additional molecules are made, the cell is now ready to go into the M phase and start specifically mitosis. So, quick recap here. We just talked about interphase only, which consists of G1, S, and G2. Remember, G1 is a period of growth. This is also where organelles get replicated. S is when DNA is duplicated or synthesized. Okay, and here I've said chromosomes are duplicated. And then finally G2, that's after DNA has been duplicated, the cell really here is making molecules to prepare for cell division, which is mitosis and cytokinesis. I do want to point out here to you, because you will need to just know these terms, and we're going to get into them more again when we talk about mitosis tomorrow. But mitosis is further divided into phases: prophase, metaphase, anaphase, and telophase or telophase. Notice that cytokinesis is treated separately. At the end of this, regardless, you end up with 2 cells. Okay, so now we're going to talk just very briefly about cell division. So cell division, as I've said several times, is mitosis and cytokinesis. Mitosis comes first. These are divided separately because mitosis is specifically and only dividing up the contents of the nucleus. So in S phase, remember the DNA got duplicated and we saw you kind of had double chromosomes. That gets separated during mitosis. So those identical sets of chromosomes we saw, they're going to get separated again during mitosis. And this just makes it so that DNA is consistent in each cell. And then that second stage, that second part of cell division is cytokinesis. And cytokinesis is treated separately because here it's just the cytoplasm and kind of all those organelles that get divided up. So that's the point at which the cells are completely separated, and they are identical at this point after cytokinesis, but you got 2 new cells. So they're treated separately. You'll see when we see them happen, it's very quick and it's hard to tell the difference, but they are divided in terms of what's being divided, they are divided in terms of what's being divided. They are classified differently in terms of what's being divided. So with mitosis, it's the nucleus and the nuclear context. Cytokinesis is the cytoplasm. Okay, so the last thing we need to get into here in terms of our objectives, as you saw, is how all of this gets regulated. So you might just think it's a lot of mumbo jumbo and it just happens magically, but it's actually back to our friends, the proteins. So we're going to have a lot of proteins that are made that are going to regulate when the cell goes into the different stages of the cell cycle. And a lot of times these proteins are going to spur other proteins to be activated. So there are 3 kind of main classes of proteins that are going to help control the cell cycle. And the first sound like they're regulating the cell cycle, which is what they do. They're called cyclins. And you can see here I've got a little graph to show you. There are several different types of cyclins. And here's an example to show you this 3 different cyclins that are upregulated or go up during these different phases of the cell cycle. So cyclin E, you'll see it go up at the beginning of this G1 to S transition, which a lot of times people call checkpoints. You'll also see an upregulation of this other one, cyclin A, at the S to G2 kind of transition, and then M phase will have a different cyclin. So each of these cyclins are going to activate different enzymes that then help the cell to proceed to the next step. So if these do not get activated, the cell cycle will not proceed. The next type are actually called internal regulatory proteins. So these are essentially inhibitors. So inhibit means to stop, and they don't let the cell cycle proceed until certain checkpoints have been reached. So like I said, people kind of call these checkpoints between them. So G1 to S, that transition, there's certain checkpoints. So for example, the cell needs to have grown in size and needs to have its organelles done. If that has not happened, it will not proceed past that point. After S, if the DNA has not been replicated, it will not proceed past that point. That doesn't happen by chance. Proteins are making sure that it does not proceed unless that checkpoint has been reached. And then finally, sometimes external factors, and this is mainly growth factors, can actually spur the cell cycle to happen. So if it's in G1, for example, growth factors can make a cell proceed to go through and eventually go through division. Cells spend a lot of time in G1, S, and G2, and growth factors actually help speed up that process because they'll activate cyclins. So again, it's all back to proteins. Proteins are helping to regulate and make sure this is timed correctly. If this gets messed up, you get cancer. That's pretty much how this goes down. So if any of these cyclins are misregulated, if things are not activated at the proper time, this can lead to uncontrolled cell growth because the cell cycle is regulating how much that cell is growing and how quickly, and the end result of that really is cancer and eventually tumor growth. So overall, that's it for today's lesson. Make sure you write your complete summary in your notes.